with the beginning of evolutionary biology, it's important that we have shared terminology so that we know that evolution is basically changes in a line of descent over time, and that's really referring to changes in the DNA, changes in the genes, changes in how the DNA is being read, and that when we're talking about over time, that's going to be species specific, that for some species like insects and mice, we can observe many, many generations. We can observe those changes in their DNA over multiple generations within one human lifespan. And that for other species like some of the trees, like the great redwoods, that we are never going to be able to observe changes in their line of descent, changes over multiple generations, because they just live so long and it takes so long to see those changes show up. So we have to know and have that shared definition, the shared definition of morphology, those physical characteristics. And then we also have to have some shared understanding of basic concepts that we'll take advantage of throughout the rest of the semester when we start to talk about case studies and basically why we're seeing what we're seeing, trying to explain the world around us. So some of those concepts will be things like looking for abiotic factors. And abiotic factors are going to be all of those non-living factors that are affecting living things. So this includes stuff like the weather, those predictable changes in temperature and precipitation over the small scale, climate, those larger scale changes, larger scale predictable patterns of temperature and precipitation, larger in scale over geographic region and over time. A lot of times when we think about abiotic factors, we think about things like fire and sunlight and water. We think about all of the rocks and the stones and all of those minerals out there, all of those non-living factors that can affect living things. And then we also need to talk about biotic factors. Now, when it comes to biotic factors, a lot of times we think about predator-prey relationships. We think about plants like this fern over here and the oxygen, that abiotic factor that's giving off that I and you and all other mammals are deeply dependent upon. We may think about creepy things like this little stink bug insect that I found walking around in my room. All of those living things that can have an effect on other living organisms. Now, another concept that we need to keep in mind when we're looking at different case studies and we're talking about things in evolutionary biology is we have to keep in mind the biomes. And when we're looking at a biome is simply defined as predictable patterns of plants and animals, flora and fauna, across a large geographic area. Now, what's making the prediction of those plants and animals in that specific geographic location will be abiotic factors like climate, like sunlight availability, like mineral and nutrient availability. So the reason why we want to keep in mind the biomes across the planet and this predictability of where we're going to see plants and animals is because this helps us to develop general rules. When we look at some of the colder regions across our planet, we know that animals and plants are going to have adaptations to deal with those colder regions. In cold regions, they're going to need to retain heat. They're going to need to retain water, interestingly enough. A lot of our colder regions are considered cold deserts because they don't get a lot of precipitation. When things do warm up to a point where you can start to run your cellular biology, you're often going to have to do it very quickly. In other words, you warm up, you got to make your babies, and then you got to prepare to go back dormant again or to die, and hope that your kids survive so that they can very quickly wake up when it warms up again, have their babies, and then they die if you happen to be a plant or even some of the animals. So we see these kinds of adaptations in the colder regions. Let's talk about the other extreme, those super hot deserts. If we have plants or animals living in a desert, what we're going to see is some of the similar types of adaptations. They're going to need to be able to grab a hold of water and keep a hold of it, but the temperature adaptations are going to have to be different. They're going to have to deal with extreme heat, and a lot of times in deserts, extreme cold as well. A lot of our deserts can have huge fluctuations between day and night. So we're going to see similar adaptations to for plants and animals in these larger biomes, these larger geographic regions. So keep in mind when we're talking about different locations on the planet, you may want to check in with that biome graph from the lecture slides to remind yourself, okay, here is where we have that tropical rainforest, consistent rainfall, consistent sunlight, consistent temperature. There's going to be a huge amount of diversity because all of our abiotic factors are consistent. Here we have close proximity to an ocean, and those ocean currents are going to be affecting everything that is happening on land. Here we have the Midwest prairie and deciduous forests, and we're going to have that fluctuation between warm temperatures in the spring and then cold temperatures in the winter. But across the seasons, we're going to see consistency in that abiotic factor of water availability. So we're going to see some adaptations. They may be similar to our extreme colder. They may be similar to some of our extreme desert adaptations. But we're going to see adaptations for that fluctuation going between those two kind of extreme situations. So always, whenever you look at a case study, anchor yourself with where this organism typically lives. Remind yourself of the surrounding abiotic and biotic factors that will be influencing that organism, and that will help give you a better understanding, and also help you to understand why we're focusing in on that organism, why we have a case study for that organism. So a couple of the things that we need to discuss. We need to focus on the idea that populations evolve. Individual organisms can change, but individual organisms are bound by their biology. There are these constraints and thresholds. We can only get so hot. We can only get so cold. We don't necessarily have the adaptations to deal with anything outside of those biological constraints. So if what we're looking for is we're looking for changes in a line of descent, well, that automatically means that we're going to have multiple generations, that we're going to have more than just one individual. We're going to need to be doing a comparison amongst all the individual organisms within a population, within a species. So please be careful here. It's very easy to slide into that misconception that an individual organism can evolve, because they can't. They can have some changes. They can pass those changes on to the next generation. But as an individual, all we can do is change. We're looking at evolution. We will be looking at populations. We'll be looking at more organisms and looking for time, looking for multiple generations. We can't just necessarily even look at one generation. We're always doing this comparison. So keep that in mind. The other thing that we need to keep in mind is that this discipline of evolutionary biology is constantly changing. We have this brief history, this brief look at how people started to explain the world around them. And some of the things I find very humorous when I look at the history of evolution is looking back at the 1600s and how people had these beautiful little diagrams of the sperm and the egg, and they thought that inside of the sperm or inside of the egg is a whole individual organism. And what happens is once conception has occurred, that organism just grows and gets bigger. The reason why they had that misconception was because they didn't really understand the gamete cells. They didn't understand that sperm and egg are haploid and that they come together, and then we have cell division that grows an organism. But inside of a little sperm and inside of a little egg, you don't have a whole adult organism that's just tiny. They did the best with the knowledge, with the data and the evidence that they could. They came to these conclusions based on the observations of the world around them. As we've gotten technology, like the microscope, to be able to see inside of these cells, we can now look at their perspective on how organisms grow and change, and we can go, wow, that's kind of wrong, and we can maybe laugh a little snarkily at them. But ultimately, we have to understand that some of the things that we have concluded right now may be just as silly to future generations, but we don't have the technology to see what's going on. We don't completely understand. We've made conclusions based on the data and evidence that we have, but we may still be wrong. So keep that in mind when you're looking at that sort of brief history of evolution. We've got people who had some initial thoughts that were incorrect. Darwin and Alfred Wallace, as we'll talk about, they too had some conclusions that were not so accurate, and as we got technology, we have revised and altered those as well, those ideas. And so we will constantly see change in this discipline of evolutionary biology. It's primarily driven by technological advancements, allowing us to be able to have a better and more refined understanding of the world around us. So we end this sort of overview of evolution and some of the basic concepts with a rule, Bergmann's rule. Now, in biology, whether it's cellular biology or environmental biology or evolutionary biology, we have lots of rules. And the thing that we have to remember about rules is that rules are great. They apply to most of the situations. But with a rule, there's usually going to be an exception. When we look at Bergmann's rules, we see that the rules are very helpful for helping us identify patterns and helping us identify different situations that seem consistent based on the external abiotic and biotic factors. But we also have to understand that there may be exceptions to those rules. So essentially with Bergmann's rule, what we see is this consistency in body shape related to temperature, related to that abiotic factor. So what do I mean by that? Well, essentially what we're getting at is this ratio of surface area to volume. If I'm a plant, like this fern, and I need to have lots and lots of interaction with the environment, I need lots of surface area. So this fern leaf has a tremendous amount of surface area. It can get lots of interaction with the environment, a lot of photosynthesis, a lot of gas exchange, carbon dioxide going in and oxygen leaving. It's very, very thin. So we have a very high surface area for this leaf and a very low volume. Now the difficulty here is when you have a high surface area, low volume, you're not going to be able to store a lot of stuff. So any organism that lives in a warm environment is going to want this high surface area, low volume situation so that they can get rid of lots of heat. But they need to balance that against not having a whole lot of water. So sometimes in dry environments, desert environments, we're going to find that they do have quite a bit of volume, but they've compromised their ability to lose heat by being able to store water. Remember, there's always exceptions to the rule. For the most part, in really warm environments, we find things that have high surface area, low volume. In the colder environments, we find a slightly different situation. In colder environments, we tend to find a very high volume, low surface area. Things in colder environments tend to be kind of round, holding all of that heat in. That's the beauty of having a high amount of volume, is that you can store lots of stuff. You can store lots of lipids, energy to burn later. One of my favorite things to do is to watch the Alaskan National Park that has a video and a grizzly bear contest. And what they do is they look at the grizzly bears in the spring, right as they come out of hibernation. And then they take video and pictures of them throughout the summer. And in September, they have what's called the Fat Bear Contest. And it's amazing. These bears pack on like 400 or 500 pounds. You see them in the beginning of the spring, they're all skinny because they used up all of their energy. And then over the course of summer, they're just storing and storing and storing all of that energy, getting big and round for the winter. They've got all the energy. They're going to hold all of that heat in that big, round, voluminous body with very low surface area. Low surface area means that they're not going to be losing as much heat to the environment. If you haven't checked it out, Google the Fat Bear Contest and see who the winners were this year. It is absolutely stunning how much energy they can pack on in such a quick amount of time. So Bergmann's rule is really looking at species response, looking at body response to the abiotic factors in the environment, and we see this pattern. More surface area, less volume, tends to be a warmer environment. You need more interaction with the environment. You don't need to store as much stuff. Lower surface area, higher volume means you tend to be storing stuff, tend to be in a colder environment. Make sure to come up with a few examples in your notebook. Share some examples with some friends. If you reach out to them through the Eagle email, make sure that you understand this concept and you know how to apply it, because these general patterns help us to understand some of the observations that we make across our planet. Now, before we leave chapter 1, the last thing that we need to do a brief review on is natural selection and genetic drift. The reason why we're going to do just a brief review now is because we're going to start applying these concepts, and then we'll get into some super nuanced detail after spring break when our semester gets a little bit harder and we start to get into more challenging concepts. But right now we want to get sort of a superficial understanding of what natural selection and genetic drift are. These are two of our main mechanisms that drive evolution. So in natural selection, we have some criteria. One of the first criteria is that individual organisms have genes. Those genes have variation. Now, the variation is going to be important. The variation could give you an advantage, could allow you to capture more resources, improve your fitness. If you're one of those organisms that has to take care of the kids after they've been born, maybe that variation helps you to take care of those kids. Regardless of what it is, some of the individuals are going to survive, but the part of natural selection that sucks is that some individuals are going to die. Only those that are successful are going to survive. And then over time, what we see is we see more consistency. We see a larger proportion of individuals in the population which have those genes. Genetic drift is a little bit different. Genetic drift takes into the part of life that just takes into effect random chance. So with genetic drift, we have some kind of random event. That random event could be something terrible like a tsunami, an earthquake, a volcano erupting. When Mount St. Helens erupted, if you were a tree on one side of the mountain and you lived, woohoo for you. But if you were on a tree on the side of the mountain that went kablooey when Mount St. Helens blew up half the mountain, it sucked to be you. You were in a pyroclastic flow. You croaked. You didn't get to pass on your genes to the next generation. And life is like that, right? We know that random things happen. You just happen to be in the wrong place at the wrong time. You get the virus. You get the parasite. You get slammed by a branch falling off the tree. You don't get to pass on your genes to the next generation. So one of the things we notice with genetic drift is that if we have a larger population, that when genetic drift happens, when we have this random event that takes out some individuals from the population, meaning they can't pass on their genes to the next generation, that when that random event happens, then in a large population, some individuals are more likely to have the rare genes or have some variation that we wouldn't necessarily find in the entire population. But if we have a very small population and a random event takes out a bunch of the individuals, then what we see going forward is more genetic similarity. Regardless of our population size, the big take-home from genetic drift is after this random event has occurred, we see a change, we see a shift in the gene frequency. Now, it's okay if these concepts seem a little bit over your head and it feels like I pushed you into the deep end. We're going to be covering these in more detail after spring break. And the reason why is because these are so fundamental to understanding evolutionary biology. When we needed to get some of these details underneath our belt, so as we start to deal with concepts and look at case studies, we can start to apply them and start to see how natural selection and genetic drift are affecting what we see around us, this diversity of life. They'll help us to explain what we are seeing and why. Our final concept that we need to touch on is this concept of artificial selection. Humans have had a huge impact on the planet. There is not a part of this planet that we haven't affected. So we have to accept our role in affecting the life around us. The example of the peppered moss that I give with genetic drift is one example of where humans have definitely affected the species and affected life around us. And what I love about that example is that it shows and really, I think, brings to the forefront how humans can be a random event, at least from the organism's point of view. So most of the time when we talk about humans having an effect on life around us, we're going to be focusing on artificial selection. And this is where humans have been selecting for very specific traits. And I can't think of an organism that best describes artificial selection than the dog. Alta, come here. So this is Alta. Alta is the combination of a Bernese Mountain Dog and a Golden Retriever. Now the Bernese Mountain Dogs were originally bred to be mountain dogs, deal with heavy snow, deal with cold temperatures, and they were bred to pull carts up to 1,000 pounds. I mean, these dogs are hugely strong. So she's got those genes inside of her. And then Golden Retrievers, well, they were really bred to be companion animals. That's why they all have such wonderfully pleasing personalities, and they don't have some of the personality problems that we see in dogs like German Shepherds, that were bred to be attack dogs, that were bred to have this very aggressive, intelligent, but we're often bred to have a much more aggressive personality, a much more assertive personality. So when we combine a Bernese Mountain Dog and a Golden Retriever, we get the best of both of those. So she's very strong. She loves to pull me when I'm cross-country skiing, doing something called skijoring. She can tolerate the cold. She loves it. And she has just this really goofy, kind of Golden Retriever happiness to her. We get the best of both of those worlds. And when we combine those two breeds, we get rid of some of the deleterious or bad genes that come along with continually inbreeding dogs to have specific characteristics. So for most of our dog species, because of inbreeding, to make them look a certain way, to have them have a certain stature, mostly physical characteristics, what we find is that we've done a lot of inbreeding, and that's led to a lot of genes that cause unfortunate things like hip dysplasia, or like in the King Charles Spaniel, with the puppies, the brain develops faster than the skull a lot of times, and so you've got this sort of ugly situation of how do you cope with that physical situation. And so we see a lot of dog people starting to make mutts, mixing up the breeds, because we need to get the genetic variation back. And that's a huge theme. Genetic variation gives a species flexibility to survive when the environment changes. We need to have genetic variation. So with our dogs, we have bred for a lot of very specific characteristics. And we're at that point with our artificial selection, and we're going back and we're having to mix it all up to try and give them less problems in their DNA. Now when I look at her, her size is the size of a wild female wolf. Dogs are, of course, related to gray wolves, and we will touch on that later this semester. They're very closely related. If she came across a male wolf in the wild, yes, if you did, and you didn't kill her, she could mate with him. I mean, we've taken that possibility away, but before we did that, she and that wolf could make babies. Well, they would be very strange-looking babies, but they could make some babies. They're still so genetically similar. Our wolves and dogs are still very much the same. So this is why when we talk about a dog is the Canis familiaris, Canis lupus familiaris. Gets 3 names. Canis is the genus, lupus is the species, and then familiaris is that subspecies, letting us know that it is still very closely related to the Canis lupus gray wolf, can still interbreed and have viable offspring. Now the other organism that we often live with is the domestic cat. Now, when it comes to the domestic cat, we have done a lot less artificial selection. And so what we find is that there is a tremendous amount of genetic variation in our domesticated cats. They are their own separate species, but they have a lot more variation. And so a lot less of those disease genes, or genes that lead to disease states, than do our domesticated dogs. So throughout this semester, we will be touching base on some of these basic concepts. We will be expanding on it by looking at different case studies. If you ever have any questions, please don't hesitate to email me. Please don't hesitate to drop into those Zoom office hours.